Cancer. A normal cell undergoes regulated division, differentiation, and apoptosis or programmed cell death. When normal cells have lost the usual control over their division, differentiation, and apoptosis, they become tumor cells. So, a tumor is the result of an abnormal proliferation of cells without differentiation or apoptosis. Tumor or neoplasm. Any abnormal proliferation of cells may be of two types: benign tumor and malignant tumor. Benign and malignant tumor. In benign tumor, neoplastic cells remain clustered together in a single mass and cannot spread to other sites. It contains cells that closely resemble normal cells and that may function like normal cells. The great majority of primary tumors arising in humans are benign and are harmless. Neoplastic cells that do not remain localized and encapsulated and become progressively invasive and malignant are described as malignant tumors. They invade surrounding normal tissues called invasiveness and spread throughout the body through circulatory or lymphatic systems called metastasis. The term cancer refers specifically to malignant tumors. Here we can see a comparison of benign and malignant tumors. Benign tumor characteristics: well differentiated, rate of growth is slow, invasiveness is absent, metastasis is absent. But malignant tumor lack differentiation, rate of growth is rapid, invasiveness and metastasis present. Both benign and malignant tumors are classified according to the type of cell from which they arise. Tumors are classified into four major groups according to their origin: epithelial, mesenchymal, hepatopoietic, and neuroectodermal. Virtually all cell types in the body can give rise to cancer, but the most common human cancers are of epithelial origin, the carcinomas. Most carcinomas fall into two categories. Squamous cell carcinomas arise from epithelia that form protective cell layers, while adenocarcinomas arise from secretory epithelia. Non-epithelial malignant tumors include sarcomas, which originate from mesenchymal cells, hematopoietic cancers, which arise from blood, and neuroectodermal tumors, which originate from components of the nervous system. Some tumors do not fit this classification system. So, carcinomas, sarcomas, neuroectodermal tumors, and hematopoietic cancers are there. Both benign and malignant tumors are classified according to the type of cell from which they arise. Tumors are classified into four major groups according to their origin, like epithelial, mesenchymal, hepatopoietic, and neuroectodermal. Virtually all cell types of the body can give rise to cancer, but the most common human cancers are of epithelial origin, the carcinomas. Most carcinomas fall into two categories. Squamous cell carcinomas arise from epithelia that form protective cell layers, while adenocarcinomas arise from secretory epithelia. Non-epithelial malignant tumors include sarcomas, which originate from mesenchymal cells. Hematopoietic cancers, which arise from blood, and neuroectodermal tumors, which originate from components of the nervous system. Some tumors do not fit this classification scheme. How many normal cells act as ancestors to form a tumor? Do the tumor cells descend from a single ancestral cell or monoclonal origin, or did a large number of normal cells undergo change, each becoming the ancestor of a distinct subpopulation of cells within a tumor mass, or polyclonal origin? Most cancers originate from single abnormal cell or monoclonal origin. Cancers are probably initiated by changes in the cell's DNA sequence, genetic changes, or changes in pattern of gene expression without a change in DNA sequence. Epigenetic changes. Epigenetic changes are nothing but genetic changes or changes in pattern of gene expression without a change in DNA sequence. Most cancers are initiated by genetic changes, and majority of them are caused by changes in somatic cells and therefore are not transmitted to the next generation. About 1% of all cancers is due to genetic changes in germinal cells and is therefore inherited. About 80% of these inherited cancers are dominant in nature. The transition of a normal cell into a tumor cell is referred to as a transformation. The transition from a normal to a transformed state is multi-step process involving genetic as well as epigenetic changes and selection of cells with the progressively increasing capacity of proliferation, invasion, and metastasis. Conceptually, this process can be divided into three distinct stages: initiation, promotion, and progression. The first step in the process is initiation. It is a process in which normal cells are changed so that they are able to form tumors. Promotion is generally associated with increased proliferation of initiated cells, which increase the population of initiated cells. Progression refers to the process of acquiring additional genetic changes that lead to malignancy and metastasis. Additional genetic changes in cancer, critical genes, are the force that drives tumorigenesis. Each successive genetic change is thought to provide the developing tumor cell with important growth advantages that allow cell clones to overgrow their more normal neighboring cells. So, initiation, genetic changes, exogenous and endogenous factors are associated with that. Then, promotion, increased cell turnover. Then, progression, genetic instability will be there. Then, metastasis, tumor invasion. Then, properties of cancer cells. Cancer cells typically display several abnormal properties as compared to normal cells that provide a description of malignancy at the cellular level, like density dependent inhibition. Normal cells in culture display density-dependent inhibition of cell proliferation and proliferate until they reach a finite cell density, which is determined partly by availability of growth factors in culture medium, but the proliferation of cancer cells is not sensitive to density-dependent inhibition. Contact inhibition. Contact inhibition. Normal cells migrate across the surface of a culture dish until they make contact with a neighboring cell. Further cell migration is then inhibited and normal cells adhere to each other, forming an orderly array of cells on the culture dish surface. This inhibition of growth after contact is called contact inhibition. Tumor cells, in contrast, continue moving after contact with their neighboring cells, migrating over adjacent cells and growing in disordered multi-layered patterns. Then, immortalization. Normal cells have a limited capacity to grow and divide both in vivo and in vitro, even if provided with optimal growth conditions. In vitro, normal cells will cease dividing after about 50 generations and then senesce and die. In contrast, malignant cells are immortal and can grow indefinitely. Invasiveness and metastasis. One of the most important characteristics of transformed cells is their invasiveness. Invasion refers to the direct migration and penetration by cancer cells into neighboring tissues. Metastasis refers to the ability of cancer cells to penetrate into lymphatic and blood vessels, circulate through the bloodstream, and then invade normal tissue elsewhere in the body. There are two properties of cancer cells that play important roles in invasion and metastasis. First, malignant cells generally secrete proteases that digest extracellular matrix components, allowing the cancer cells to invade adjacent normal tissues. Second, cancer cells also secrete growth factors that promote the formation of new blood vessels. Angiogenesis. Angiogenesis is needed to support the growth of a tumor by supplying oxygen and nutrients to the proliferating tumor cells. Then, loss of anchorage dependence. Most normal cells must be attached to a rigid substratum. They must be anchored in order to grow. Transformed cells can grow even when they are not attached to the substratum, as for example when they are suspended in a semi-solid medium containing agar or methyl cellulose. Lower growth factor requirements. Normal cells grow in culture require various growth factors to continue proliferating. In normal human tissues, growth factors are generally produced extracellularly at distant sites and then either carried through the bloodstream or diffused to their nearby target cells. However, tumor cells often produce their own growth factors that bind to and stimulate the activity of receptors that are also present on the same tumor cell that are producing the growth factor. This results in a continuous self-generated proliferative signal known as autocrine stimulation that drives proliferation of the tumor cells continuously even in the absence of any exogenous proliferative signal. Autocrine stimulation is manifested as a reduced requirement for serum because serum is the source of many of the growth factors in the media used to propagate cells in vitro. Failed to undergo apoptosis. Another general characteristic of most cancer cells is that they fail to undergo apoptosis and therefore exhibit increased lifespans compared to their normal counterparts. This failure of cancer cells to undergo apoptosis contributes substantially to tumor development. Cancer cells are often characterized by increased resistance to apoptosis, allowing their survival under abnormal growth stimulation. Now we are going to learn about the major topic, the molecular basis of cancer. All genes whose genetic and epigenetic changes contribute to the causation of cancer is described as cancer critical genes. These genes contribute to the transformation process by driving cell proliferation or reducing sensitivity to cell death and differentiation. These genes may be involved in the following processes like cell cycle progression, RB1 MYC, differentiation process like APC, DNA repair ATM and BRCA, cell death BCL2. Cancer critical genes are grouped into two broad classes according to whether the cancer risk arises from a gain of function mutation of a gene or a loss of function mutation. Genes for which a gain of function mutation drives a cell toward cancer are called proto-oncogenes, whereas genes for which loss of function mutation creates the cancer are called tumor suppressor genes. Mutation of a cancer critical gene can be either dominant or recessive. A gain in function mutation in a single copy of a proto-oncogene can drive a cell toward the cancer. On the other hand, the cancer-causing mutations of tumor suppressor genes are generally recessive. It means both copies of normal gene must be removed or inactivated in the diploid somatic cell before an effect is seen. So mutation of a cancer critical gene can be either dominant or recessive. A gain in function mutation of a single copy of a proto-oncogene can drive a cell towards the cancer. On the other hand, the cancer-causing mutations of tumor suppressor genes are generally recessive. It means both copies of the normal gene must be removed or inactivated in the diploid somatic cell before an effect is seen. Then proto-oncogenes. Proto-oncogenes control the growth and division of cells. The proteins encoded by proto-oncogenes induce growth factor receptors, transcription factors, and signal transducers. So, proto-oncogenes control the growth and division of cells. The proteins encoded by proto-oncogenes include growth factor receptors, transcription factors, and signal transducers. They contribute to the transformation process by driving cell proliferation or reducing sensitivity to cell death. Then there are many examples of proto-oncogenes and their products like SIS, synthesis of platelet-derived growth factor. Then ERBB, synthesis of epidermal growth factor receptor. Then SRC, synthesis of tyrosine kinase. ABL, synthesis of tyrosine kinase. Then KRAS, synthesis of GTPase. GEN, synthesis of transcription factors. FOS, synthesis of transcription factors. MYC, synthesis of transcription factors. SRC, synthesis of tyrosine kinase. So these are proto-oncogenes and their products. Synthesis of tyrosine kinase, then GEN, synthesis of transcription factors. FOS, synthesis of transcription factors. MYC, synthesis of transcription factors. These are proto-oncogenes. Today, more than 40 different human proto-oncogenes are known. Several types of genetic and epigenetic changes convert these proto-oncogenes into oncogenes. Oncogenes arise as a result of changes that increase the expression level or activity of a proto-oncogene. Underlying mechanisms associated with proto-oncogene activation include the following, like point mutation. A proto-oncogene may be converted into an oncogene through a single alteration of a nucleotide. This alteration may be the deletion of a base, the insertion of an extra base, or the substitution of one base for another. However, regardless of the type or cause of such a mutation, it usually changes the amino acid sequence of the encoded protein and thus alters protein function. A point mutation can increase gene function or it can interrupt gene function. An example of in which point mutation converts proto-oncogene to oncogene is reported by the RAS proto-oncogene comprising HRAS, NRAS, and KRAS. K5. Then chromosomal translocations. Chromosomal translocations may result in activation of proto-oncogene. Translocation-induced overexpression of a proto-oncogene is best exemplified by Burkitt's lymphoma. It is a childhood tumor, particularly affecting the jaw and found mainly in Africa. In Burkitt's lymphoma, the MYC-containing segments of chromosome 8q24 translocate to chromosome 14q32. This translocation places MYC gene close to the Ig heavy chain gene. The translocated MYC gene, here regulated by the Ig gene enhancer, is continuously expressed, causing cell to become cancerous. The product of MYC gene acts as a transcription factor that affects diverse cellular processes involved in cell growth, proliferation, apoptosis, and cellular metabolism. Hence, altered expression of MYC gene leads to deregulation of a large number of target genes. Then similarly, patients with chronic myelogenous leukemia, also called chronic granulocytic leukemia, contain an abnormal Philadelphia chromosome, which forms as a result of balanced reciprocal 922 translocation. This translocation creates a novel chimeric BCR-ABL gene present on chromosome 9 and exchange transfers the ABL gene to chromosome 22, known as Philadelphia chromosome. ABL is a proto-oncogene and encodes a tyrosine kinase. ABL stands for Abelson, the name of leukemia virus which carries similar protein. The BCR gene codes for BCR protein whose function is not clear. Chimeric BCR-ABL gene codes fusion protein of 185 to 230 K Dalton, depending on the breakpoint in the BCR and ABL gene. In contrast to the normal ABL protein, BCR-ABL fusion protein exhibit constitutively active tyrosine kinase activity. Here we could see a picture of generation of a chimeric BCR-ABL gene by chromosome translocation. The CABL gene in a normal cell is located in chromosome 9 and encodes a tyrosine kinase. During malignant transformation of myeloid cells, a portion of chromosome 9 that contains the ABL locus translocates to chromosome 22 at the breakpoint cluster region BCR gene and generates the chimeric BCR-ABL gene. So normal chromosome is here. Normal chromosome, another one is here. Chromosome break is happening, then translocation is happening. It forms a Philadelphia chromosome. BCR gene is there, ABL gene is associated with that. So then examples of specific chromosome aberrations and associated cancer chromosome alteration, chronic myelogenous leukemia translocation, acute myelogenous leukemia, retinoblastoma, Wilms tumor. It happens, deletion and some translocations. Then gene amplification. Activation of proto-oncogenes results from reduplication and amplification of other DNA sequences. The most interesting case of amplification involved NMYC in neuroblastoma. Gene amplification is usually an unstable genetic condition that can only be maintained under strong selective pressure. Then insertional activation. Insertion of mobile genetic elements such as retrovirus changes the expression of cancer but leave their coding sequence unaltered. The best characterized is CMYZ, whose expression is elevated after the insertion of a non-defective retrovirus in vicinity of the gene. Avian leukosis virus ALV is a retrovirus that does not carry any viral oncogene, yet is able to transform B cells into lymphomas. This particular retrovirus has been shown to integrate within the MYC proto-oncogene, which contains 3 exons. Exon 1 of MYC gene has an unknown function. Exons 2 and 3 encode the MYC protein. Insertion of ALV between exon 1 and exon 2 has been shown in some cases to allow the previous promoter to increase transcription of exons 2 and 3, resulting in increased synthesis of MYC. Then exon 1, exon 2, exon 3, etc. are there. CMYZ proto-oncogene is there. ALV integration, exon 1, LTR regions, exon 2, exon 3. Relation of DNA sequence is there. Then exon 2 is there, exon 3, exon 2, exon 3. Transcription and splicing of CMYZ genome. Then activation of the MYC oncogene as a sequence of ALV integration. In a normal cell, the 3 exons of the MYC gene are normally spliced into an mRNA that can be translated into the MYC protein. Only exons 2 and 3 actually code for the protein, and ALV integrates into the MYC locus, usually between exons 1 and 2. The presence of the LTR sequence in the transcript results in increased expression of the MYC protein, which is harmful to the cell. The integration of ALV is often followed by deletion of portions of viral sequence, leaving one of the LTRs intact. Then next topic, tumor suppressor genes. Tumor suppressor genes can directly or indirectly inhibit cell growth. Those that directly inhibit cell growth or promote cell death are known as gatekeepers, and their activity is rate limiting or for tumor cell proliferation. Those tumor suppressor genes that do not directly suppress proliferation but function to promote genetic stability are known as caretakers. Caretakers function in DNA repair pathway, and elimination of caretakers results in increased mutation rates. 5 broad classes of genes are generally grouped into tumor suppressor genes: genes that regulate or inhibit cell cycle progression, P16 and RB1; genes that encode receptors or developmental signals that inhibit cell proliferation, or the Hedgehog receptor; genes that regulate or inhibit cell cycle progression. Then some other genes that encode receptors or developmental signals that inhibit cell proliferation. Then genes encoding checkpoint control proteins that arrest the cell cycle. Then genes that promote apoptosis. Then genes that encode enzymes that participate in DNA repair. Example of tumor suppressor genes and their functions: tumor suppressor genes BRCA1 and BRCA2, transcription factor and DNA repair. Then HMLH1, DNA mismatch pair; NF1, GTPase; P53, transcription factor; RB1, cell cycle checkpoint. P53 is well-studied tumor suppressor gene that encodes a polypeptide which acts as a transcription factor. Mutations in the P53 gene are associated with more than 50% of human cancers. P53 protein plays an important role in the maintenance of genetic stability. Loss of P53 abolishes the DNA damage checkpoint. Cells with functional P53 become arrested in G1 when exposed to DNA damaging radiation, whereas cells lacking functional P53 do not. This permits the cells to repair the DNA damage prior to stages of the fixation and propagation, which may lead to tumor formation. Moreover, P53 was shown to facilitate general genomic repair of DNA damage and to bind proteins involved in DNA repair. On the other hand, in many cellular systems, P53 promotes apoptosis of cells. Apoptosis of cells. Harboring irreparable damaged DNA. The active form of P53 is a tetramer of 4 identical subunits. A missense point mutation in one of the 2 P53 alleles in a cell can abrogate almost all P53 activity, because virtually all the oligomers will contain at least one defective subunit, and such oligomers have reduced ability to activate transcription. Oncogenic P53 mutations thus act as dominant negatives, with mutations in a single allele causing a loss of function. Dominant negative mutations can occur in proteins whose active forms are multimeric, or those function depends on interactions with other proteins. In contrast, loss of function mutations in other tumor suppressor genes such as RB1 are recessive because the encoded protein function is monomers, and mutation of a single allele has little functional consequence effect. Then BRCA1, breast cancer susceptibility gene 1. Breast cancer susceptibility gene 1 and BRCA2 are tumor suppressor genes. Both genes contribute to checkpoint control of cell cycle, DNA repair, and transcriptional regulation in response to DNA damage. Mutation of these genes has been linked to hereditary breast and ovarian cancer. Alternation of genes involved in the repair of damaged DNA are also responsible for a number of human cancers. In view of thousands of damage events that genomes suffer every day, coupled with the errors that occur when the genome replicates, it is essential that cells possess efficient repair systems. Without these repair systems, a genome could not be able to maintain its essential cellular functions for more than a few hours before key genes become inactivated by DNA damage. Deficiency in DNA repair causes several human diseases. One of the best characterized of this is Xeroderma pigmentosum, which results from a mutation in any one of several genes for proteins involved in nucleotide excision repair. Apoptotic genes can function as proto-oncogenes or tumor suppressor genes. Anti-apoptotic genes such as BCL2 behave as proto-oncogenes because overproduction of their encoded protein prevent normal apoptosis. Conversely, apoptotic genes whose protein products stimulate apoptosis behave as tumor suppressors. Then carcinogen. Agents that initiate or promote tumor formation are called carcinogens. Three classes of carcinogenic agents are known: radiations like UV radiation and gamma ray, chemicals like benzopyrene and benzene, and biological oncovirus agents. The ability of chemical and physical carcinogens to induce cancer can be accounted for by the DNA damage that they cause and by the errors introduced into DNA during the cell's effort to repair the damage. Chemical carcinogens have a very broad range of structures that have little or no obvious structure or biochemical similarity with each other. There are two types of chemical carcinogens. One called direct acting carcinogens that act directly without any metabolic activation, and the other called indirect acting carcinogens which require metabolic activation for them to act as carcinogens. Aflatoxin, a mycotoxin, is an example of indirect acting carcinogen. It is activated into alpha toxin 2,3 epoxide by the action of intracellular enzymes, which is associated with characteristic mutations of the p53 gene. Aflatoxins produced by the fungi Aspergillus flavus and Aspergillus parasiticus are among the most potent liver carcinogens known. Chemically, the aflatoxins consist of a diurofuran ring system fused to a constituted coumarin moiety with a methoxy group attached at the corresponding benzene ring. After chemical modifications by the enzyme, aflatoxin becomes linked to G residues in DNA and induces G to T transversions. Other common examples of chemical carcinogens which are associated with a number of human cancers are benzene, leukemia, arsenic, lung and skin, cadmium, prostate, radon, lung, asbestos, lung and gastrointestinal tract, and vinyl chloride, angiosarcoma, liver. Some carcinogens contribute to cancer development by stimulating cancer proliferation rather than by inducing mutations. Such components are referred to as tumor promoters. 4-bol esters that stimulate cell proliferation by activating protein kinase C are classic examples which stimulate proliferation of the mutated cells. Then retinoblastoma. Retinoblastoma is a childhood tumor of the retina. The gene whose loss is critical for development of the cancer is the RB1 gene. Retinoblastoma arises from when both copies of RB1 gene is inactivated. There are two forms of the disease, one hereditary and one sporadic. In the hereditary retinoblastoma or familial form, individuals inherit a single defective copy of RB1 gene, and tumors occur when the remaining normal copy is lost or inactivated by a somatic mutation. In this instance, there are usually multiple foci of tumors arising in both eyes and is therefore called bilateral retinoblastoma. In the non-hereditary retinoblastoma or a sporadic form, all cells initially contain two functional copies of the RB1 gene, and the tumor arises because both copies are lost or inactivated through the coincidence of two somatic mutations in one cell. Because of this is an unlikely occurrence, sporadic retinoblastoma is rare, develops late in life, and usually affects only one eye. Mutant RB1 gene is the familial retinoblastoma. Genotype of fertilized egg, sporadic retinoblastoma happens. Then first somatic mutation. After that, 2 mutant RB1 genes are there, then mutant RB1 gene, second somatic mutation that will lead to 2 mutant RB1 gene. In familial retinoblastoma, the zygote fertilized egg from which a child derives carries one defective copy of the RB1 gene, and all retinal cells in this child will therefore carry only a single functional RB1 gene copy. If the surviving copy of the RB1 gene is eliminated in a retinal cell by a somatic mutation, that cell will lack all RB1 gene function and will be poisoned to proliferate into a mass of tumor cells. In sporadic retinoblastoma, the zygote is genetically wild type at the RB1 locus. In the retina of a resulting child, retinoblastoma development will require 2 successive somatic mutations, striking the 2 copies of RB1 gene carried by a lineage of retinal precursor cells, yielding once again the same outcome, a cell poisoned to proliferate into a tumor mass. Because only a single somatic mutation is needed to eliminate RB1 function in familial cases, multiple cells in both eyes are affected. However, the 2 somatic mutations required in sporadic diseases are unlikely to affect a single cell lineage, yielding at most one tumor. Then, oncoviruses or tumor viruses. The transforming activity of a tumor virus residing in a particular oncogene or oncogenes is carried in the viral genome. Oncogenes were given their name by virtue of their ability to convert normal cells to an oncogenic state. Oncovirus containing viral oncogenes may be DNA containing oncoviruses like hepatitis B virus, herpes virus, and SV40 virus carry oncogenes without cellular counterparts. These oncogenes may work by inhibiting the activity of the cellular tumor suppressor. DNA tumor viruses act mainly by interfering with cell cycle controls. For example, papillomavirus causes human warts and carcinomas of the uterine cervix, uses 2 viral proteins E6 and E7 to sequester the host cells P53 and RP respectively. Similarly, EIA and EIB proteins of adenovirus inactivated the RB and P53 tumor suppressor proteins with E1A binding to RB and EIB binding to P53. Some of the example of DNA containing oncovirus that cause human cancer are hepatitis B virus, liver cancer, papillomavirus, cervical and other anogenital cancers, then Epstein-Barr virus, Burkitt's lymphoma and nasopharyngeal carcinoma, and Kaposi's sarcoma associated herpes virus, Kaposi's sarcoma. RNA containing oncoviruses such as transforming retrovirus contain oncogenes. Viral oncogenes were first reported in Rous sarcoma virus by Peyton Rous in 1911, which transforms chicken embryo fibroblasts in culture and induces sarcoma. The first oncogene originally found in the Rous sarcoma virus was designated as the SRC oncogene. Some of the examples of RNA containing oncoviruses that cause human cancer are human T cell lymphotrophic virus, HTLV1, adult T cell leukemia lymphoma, HTLV2, hairy cell leukemia, and simian virus 40, SV40, non-Hodgkin's lymphoma. Then next topic, retroviral oncogenes. American microbiologist H. Warmus and H. Bishop in 1976 demonstrated that normal chicken cells contain genes that are closely related to the retroviral SRC oncogenes codes for SRC protein tyrosine kinase and Rous sarcoma virus. They published the remarkable conclusion that the oncogene in the virus did not represent a true viral gene, but was a normal cellular gene with which the virus had acquired during replication in the host cell and therefore carried along. SRC related sequences are also found in normal DNA of a wide range of vertebrates, including human. Until now, more than 40 different oncogenes have been demonstrated. The normal cell genes from which the retroviral oncogenes originate are called proto-oncogenes. In 1972, R.J. Hubner and C.J. Todaro suggested that mutations or genetic rearrangements of proto-oncogenes by carcinogens or viruses might alter the normally regulated function of these genes, converting them into potent cancer causing oncogenes. The viral oncogenes and their cellular counterparts are described by using prefixes V4 viral and C4 cellular. So the oncogene carried by Rho-sarcoma virus is called VSRC, and the proto-oncogene related to its cellular genome is called CSRC. So when we look at this picture, you could see normal cells are represented. Proto-oncogenes are there associated with the synthesis of growth factor and growth factor receptor, signal transducers, transcription factors. Then we just form viral oncogenes are there, cellular oncogenes are there, transformed cells are there. Qualitatively and quantitatively altered expression of proteins is also happening. So that's all about this topic. Thank you for listening.